The oncology pillar is part of the Center for Medicines Discovery, reflecting its drug discovery engine in oncology. To be able to find new medicines to treat cancer patients, we take two approaches. One is that we take advantage of deep in-house knowledge on key signal transduction and other molecular processes that control cancer growth, as well as their interaction between cancer cells and immune cells. Often these processes are controlled by key molecular switches. We refer to them as post-translational modifications, such as ubiquitination or phosphorylation and others, which we understand in great detail and can figure out which of those may reflect Achilles' heels for interfering with cancer cell growth.
Our second approach is to use state-of-the-art platforms to explore drug targets and accelerate discovery further. So we'll look further into the human cancer genome and also the proteome. We also perform regularly genotype screening, phenotypic screening and drug screening. And we also have great expertise in structure-based drug discovery, or XChem, to identify novel drug candidates.
Glioblastoma is a cancer of unmet clinical need, where the standard of care therapy has not advanced for the last 20 years. This lack of therapeutic advancement requires substantial innovation, including the application of novel, cutting-edge technologies and new, more predictive cancer models that more closely mimic the disease biology, followed by the application of novel therapeutic modalities, for example, small molecule drugs or immunotherapeutics. This is key if new, actionable drug targets or therapeutics are to be identified and developed into the next line of glioblastoma treatments.
Spatial proteomics is a very useful tool to detect the spatial distribution of novel biomarkers and drug targets within the three-dimensional space of tumor tissue and the tumor microenvironment.
We're part of a large international multidisciplinary consortium called PROTECT. It's funded by the prestigious Cancer Grand Challenges, and our focus is childhood cancers, and we want to deploy degraded technologies against these because many of the drivers of childhood cancers are not really tractable by traditional small molecule approaches. Here at the CMD, we are using state-of-the-art high-throughput crystallography to pinpoint particular opportunities for designing degraders. We are using the XChem facility at Diamond Light Source. This is a facility that I developed over the last decade to do crystallographic fragment screening against these targets.
Our lab is dedicated to discovering new therapeutic options in immuno-oncology. Specifically, we focus on understanding how cancer cells prevent detection by the immune system through a set of mechanisms collectively known as cancer immune evasion. One key aspect of our research is investigating the role of innate immunity, the body's first line of defense against pathogens. It has been observed that cancer cells can manipulate these innate immune pathways to avoid immune destruction. In this context, we have identified a family of enzymes called the acylating enzymes as potential regulators of cancer immune evasion. Given their role in modulating immune responses, we believe these enzymes could serve as promising immunotherapeutic targets.
The immuno-oncology pillar in the CMD sits right at the interface between academia and pharma. We are able to facilitate collaborations between academia and industry, and we can create fertile ground for innovation and spin-outs.
All these new insights create great opportunities to partner up with pharma, who can help accelerating drug development and take them through the different clinical trial stages, all the way to benefit patients.